going to present it. So, biodiversity. See, I'm not going in detail about each small event, but overall, I'm going to complete the entire syllabus. There's a first unit and fourth unit also, if the time permits, I will try to cover. But first year, I will first semester. First unit definitely I'll be doing in detail. So, what is biodiversity basically? See, we are having the paper specialization microbial biodiversity. So diversity in the sense variance or variety or changes. So the variance or variety among the living organisms is called as biodiversity. Okay, the bio is life, diversity is variety or variance or the change, that is called as variety. So biodiversity is the variety or the variance among so that is one definition. The biodiversity is a variety of life variations among the living organisms on the earth, and there is an essential interdependence. Interdependence among the living organisms is called as biodiversity. There is one way. There is second. This is a more acceptable definition, which is proposed in like a summit of biodiversity in Rio, Brazil in 1992. And it has been approved by almost 154 countries. Here, this definition, the variability among the living organisms from all sources, including there's inter alia, terrestrial, marine, and all aquatic, other aquatic ecosystems. In the sense, all ecosystems, whether it is a water, terrestrial, air, what different types of ecosystem complexes of which the area part, this include diversity within the species, between the species, and the ecosystem is called as biodiversity. Whatever the ecosystem is there in it, whatever the variations among the species, between the species, and ecosystem that is called as biodiversity. This is the definition given by the Biodiversity Summit in 1992. So first definition, as I told you, what I follow. So you follow this one. This is more than enough. Biodiversity is a variety of life on the earth and the essential interdependence of all living things. So definitely you will be asked about the biodiversity, otherwise different types of biodiversity. So this definition is mandatory for you. So almost 1.4 million species are there, which are characterized, and many tens of millions of species are yet unknown. There are many things which are not yet characterized, which are not yet known. Okay, so there are many things are there. So in the biodiversity, why we'll see about why we should study and other things. See, different forms of biodiversity we'll see. Basically, it is divided into three groups. There is genetic biodiversity, species biodiversity, and ecosystem biodiversity. Genetic biodiversity, if the species is also same, but genetical divergence is there. Due to genetical divergence or due to genetical changes within a species, if the organisms are being changed, this is called as genetical biodiversity. Within the species, if the organisms are showing different phenotypes because of the genetical alteration of genetical makeup, it is called as genetical biodiversity. See, here the dogs are there, here the species is also same, but due to differential genetical makeup, they are looking into different forms. Phenotypes are different here. That's what it is, a genetical biodiversity. And see here the patterns of the shells, and these are the dogs and the rats also. You must have seen different types of rats are there. So all these here, the species is the same, but a bit of DNA is getting modified here. That's what it is called as genetic biodiversity. And species biodiversity, here the species or genuses all put together gets changed. So the different forms of life which are staying in that area, one area, is, we can call it as a species biodiversity. Monkeys, golden skimmers, there's a different meadow beauty plant, different things which all put together, they belongs to different genus as well as species. That is called as species biodiversity. And see here, this beetles comes under the genetical biodiversity. When you compare beetles versus donkeys and monkeys, it is going to be a species biodiversity. So third, ecosystem biodiversity. What do you mean by ecosystem? Basically, the living organisms of an area in connection with its physical and chemical environment makes an ecosystem. If you take the pond, in that pond you are having living organisms. There's animals, plants, microbes, and like mushrooms. Sometimes even fungi also may also be there. So actinomycetes, all these microbes are there, and plants are there. Some fishes are also there in that one. These are all living organisms. And apart from that, you are having non-living organisms. The physical component is water, and whatever the pond is there, that is making the, so the physical environment plus chemical environment, the pH of that pond, or temperature, all these things put together makes an ecosystem. So ecosystems here, see, different types of ecosystems are there. Rocky ecosystem, sand dune ecosystem, estuary ecosystem. Estuary is the area where the fresh water mixes with the rivers, where they mixes with oceans. It is a connecting point between there's a fresh water as well as there's a marine water system, and salt marshes, coral reefs, and all these things are ecosystems. And here the freshwater ecosystems are lakes, ponds, rivers, and remaining are the marine ecosystems. Even these desert ecosystems are there, mountain ecosystems are there. even snow, the Arctic ecosystem is there, all different types of are there. So here, if you see in biodiversity, see variations among the living organisms and their interdependence is there, it should be there in a biodiversity. And it is there at 3 level. One is genetical level. Genetical level, species also same. Species is also same, but there is a change in genetical makeup. Genetical makeup. See, maybe some genes must have got mutated. You must be aware the coat gene, the color gene is regulated by multiple alleles. If you take even human hair gene also controlled by the multiple alleles, and even this cows and dogs, here the color is coded by the multiple alleles. So here what happened, due to mutations or recombination, sometimes some alleles may be missed, or maybe or maybe some alleles may be more than 1 or 2 copies. So this may lead into different forms. So here the species is same, but only due to alteration in one gene or maybe few genes, here it is getting changed. So it is called as genetical biodiversity. It is called as genetical biodiversity. And species biodiversity. Actually, here species biodiversity, it starts from the species. It is not limited to the species, but it starts from this species. Like dog, this cat and lion. Many such kind of things where the genus is same but species is different. Here the level for species biodiversity, minimum change variation should be at species level, and it could be genus and phylums also. See this the flower plant belongs to different phyla, and this monkey belongs to different phyla, and this skimmer belongs to different phyla. So minimum unit that require for the species biodiversity is at least a difference should be there in the species level. Hope you got understood. Third, ecosystem biodiversity. Here, ecosystem, ecosystem is nothing but the living organism along with its physical and chemical components of an area is called as ecosystem. And ecosystem makes a unique system. See, pond ecosystem is different from the desert ecosystem, and the rocky mountain ecosystem is different from rocky coastal ecosystem. Rocky coast. See, both the areas, the rocks are there, but rocky mountain is a dry area. Rocky coastal is a completely moistured or water area. So these are the 3 ecosystems. See, this is ecosystem biodiversity. This is a pond ecosystem. This is a tropical forest ecosystem, and more biodiversity is there in case of your tropical forest. And this is the coral reefs. The third one is a coral reefs. So these are the different types of biodiversities we have. And if you see the pyramid, if you arrange them in number pyramid, genetical biodiversity is going to be very high. Species biodiversity is moderate, whereas ecosystem biodiversity is the minimum. So if you number wise, if you arrange 3 types of biodiversities, then the genetic biodiversity will be at the base. And see, because there are many species, and among the species there are many strains, and because of the mutations, single species got multiple types of variants. So genetic biodiversity is going to be huge. Whereas ecosystem, we can name few on your fingers. This biodiversity, see, pond ecosystem and desert ecosystem. Like there are very few ecosystems, so it will be there at the top. In the ecosystem again, you are having some other component. Habitat. You should know these terms. Habitat is the area where actually the organism survives. It is the physical and chemical description where the creature lives. where, see, if this is the habitat, this is a rocky forest, a rocky mountain, sorry. This is a rocky mountain or rocky forest also, you can say rocky mountain, rocky forest. So here, because rocks are there as well as trees are also there, rocky forest. So in that one, where does your rat or rodent survives? Is it near the cactus, or is it near the that tree, or is it near the stone, or is it near the this bushes? Where does it survive? That is uh, the habitat. And sometimes based on are also given. See the Arctic Canada. See, sometimes we'll be giving the name Arctic panda or Himalayan lion or Himalayan cow, something like this. And here there is a one more habitat is it is a dominant vegetation of the place where the creature lives. That's what and it also gives the where the creature lives. And next, coming to the species. Species, see, all these three are same species. 3, 4 are there here. 4 dogs are there. All of them are same species. Here, species can be defined a group of morphologically similar creatures. Like in appearance, they should be similar. All the creatures should be similar, with the condition of they should interbreed to produce a fertile offspring. See, the mule may appear similar to donkey sometime, but it cannot interbreed with. Sometimes you see, donkey and horse may appear physically similar, morphologically it may appear similar, but they cannot interbreed to produce a fertile offspring. Mule is produced and which is a sterile, which is not a fertile thing. So, a species is a group of similar organisms, morphologically similar organisms, which can freely interbreed to give a fertile offspring. Okay. Otherwise, we can call them as a reproductively isolated. They can reproduce. Only these group of people or these group of organisms can freely reproduce. They are separated. Okay. And see, this definition we are having some problems like extinct creatures. We have given some groups to them, and see, so bacteria, it cannot do a sexual reproduction. Actually, the definition should be a sexual reproduction should be there. So these are and sometimes some like man versus chimp testing may not be ethically correct. Okay. The other way, the second way of it is based on similar DNA, similarity DNA. How much the DNA is matching in between the man and man? Almost it is going to match 99%. It's 99% only, not 100%. Even two identical twins also does not shows the 100%. See, one DNA, what you get from your parents is something, and second, see, environmental exposure brings the changes in the DNA. Some mutations, some alterations do happen. So, some extent of mutations, some extent of DNA change, spontaneous mutations are also possible when the DNA replication goes on. When the DNA replication goes on, some wrong bases are also added in that. So there is every possibility of having not 100% similar. So don't expect 100% similarity there, but there should be a maximum match. So it is believed that the monkeys, apes are closely associated with man. And orangutan, there is a orangutan who can walk, and this is a monkey ape who can walk similar to man. And he's in fact having a brain of thinking and reasoning, and he's having almost 97 to 98% DNA similar to man. 97%, and remaining apes 95% monkeys they are having 95% DNA match. So if you take as this match decreases, the relatedness will be also decreases. If you are taking donkey, the matching will be somewhere around 70-75%. So you are taking the DNA sequence of man and you are taking the DNA sequence for somebody else and you will be comparing. Here we'll be collecting the sequences and see which sequence we should compare. There is a question mark here. Which sequence? See, there is a huge amount of DNA in case of man, and you take the other organisms also. You are having a lot of DNA. So what DNA should be compared? In case of bacteria, generally we will compare 16S RNA. It is a ribosomal RNA. It is a 16S RNA we are supposed to compare. Whereas in case of eukaryotes, earlier we used to compare 18S RNA and IS element. IS elements. These two used to be done. And even that is being practiced for the eukaryotic microbes. Eukaryotic microbes like fungi, yeast. For this kind of even algae, small organisms, we can compare this 18S RNA as well as IS elements. Whereas for higher organisms, for human being, I want to compare monkey, donkey, man. I want to compare these three. So 18S RNA may not be that much fruitful. So the best thing what we can do is cytochrome. There's a cytochrome. See, we need to take a gene which is conserved, highly conserved. So a little bit modification in that one will give you the indication that the probable evolution. So cytochrome C. Cytochrome C is considered to be one of the more conserved sequence because it is required for the energy production process. Oxidation reduction process it is required. So it is a highly conserved. So we take cytochrome C as a reference standard and we will compare cytochrome C. If it is highly matching, then we can say that it is closely related. If it is having very less matching, then we can call it as a non-related or less related kind of thing. So this is how we compare the DNA sequences. There are other methods also like blood proteins. RH factor is there in man as well as monkeys also. RH factor, and in fact, it was discovered first in monkeys, rhesus monkeys, later in man. And this even blood grouping factors are also there in orangutan. Similar ABO system is there in orangutan. So and biochemistry like enzymes, cooking of enzymes, and embryology. You must have seen embryological evidences. So whatever maybe it will. First, zygote it should start, so unicellular from it starts, then blastula, there's the prolifera characteristics, and then gastrula, cylindrics property, and then you will be having fishes properties, all, see, one by one, at the end it will acquire the characteristics of man. See, this is based on the DNA profiling percentage. Among the primates, almost 93% similarities there man to primates. Human shares 98 to 99% of their DNA with chimps. This is how much the differences are there among different organisms. So what are the values of biodiversity? Why we should study about why the people are worried about biodiversity? Huge, and it has been started as a course, and I hope you people have also joined some biodiversity course which is being offered by DST. And why that's much importance is there for biodiversity? Because of COVID situation, we came to know the value of oxygen, and where biodiversity, the plants of this earth, are giving huge amount of oxygen. We never know. We never bothered about it. We never cared about it. But once, let's see, this COVID situation has realized us actually the value of this biodiversity of plants for oxygen production. So it is biodiversity having intrinsic value as well as extrinsic value. Intrinsic value, within, if something is having a value of within, it is called as intrinsic value. So something, if it is having external value, it is called as extrinsic value. If it is something giving out, that is called as extrinsic. Something of its own, it is called as intrinsic values. and something utilitarian value, something that is useful, something of biodiversity. It is useful. Some you will use it for yourself, or you will sell it for money. That is called as utilitarian value. So utilitarian values, like you will be getting a lot of goods, sustainability. But see, first, what is this intrinsic value? What is the intrinsic value you are having? Huh? What is intrinsic value? It is aesthetic. It's recreation. You are able to create a recreation, beauty, because of the plantation. There will be a aesthetic value created over there. Gardens are created over there. You can go and you can sit over there and you can leave your pain or whatever the thing for some time. So that is intrinsic value. And extrinsic value or utilitarian value is the one which you can use and you can give it for somebody for money or services kind of thing. Like two examples are given. Like sustainable, you are getting timber, you are getting turmeric. Not one, many, all the products you are getting from them. All Ayurvedic compounds you are getting from there. And ecological services you are getting, rain, whatever the rains you are getting, and you are getting recycling of materials, nutritional cycles you are getting, nitrogen cycle, carbon cycle, all those things. That is and information, national parks and wooden gardens that we are maintaining. So, oxygen. Now we realize the importance of oxygen, how much the oxygen is important, and how much these plants are producing. And algae also, ocean algae, this marine algae, actually they are contributing huge for this oxygen production. And tropical forests, actually, this Amazon forest, which are located in Africa, they are contributing huge for this oxygen production. So, second, there's food production. See, whatever the food that you are see, we are getting the plants from the forest, from the wild areas, and we are cultivating them. Even though you are growing the paddy in your fields, that is also one biodiversity. That is also one of the biodiversity. So, in water also you are getting, whether it is by rains or underground, and the medicines you are getting, aesthetics, there is a appearance. See, here we have clubbed intrinsic and extrinsic, both the things we have, just everything that is explained over here. And ideas, what ideas will you get from biodiversity? What ideas will you get? See, the big engineering constructions which are there, they are constructed based on the designs of leaves, designs of the nests which are created by the insects and some birds. Many engineering construction, nature is a beautiful engineer. Nature is a beautiful engineer. So, based on the observations which are there in the nature, we are getting a lot of ideas. And even several chemical reactions which are taking place in the nature, by taking them, we are constructing large scale reactions. So, applications, if they are coming, you have to expand these things. And see, further to go, consumptive values, there's food and drink we are getting. See, vinegar, honey we are getting, vinegar we are getting as a drink purpose. And even there's sometimes natural fermented toddy and other things also we are getting. Fuels we are getting is in the form of coal, in the form of wood, in the form of ethanol we are getting from the environment or biodiversity. Medicines, Ayurvedic, and see, for the allopathic medicines also, aspirin if you want to produce, for aspirin you require a lot of solvents, a lot of precursors which are coming from the nature itself. So for medicines also, we require the biodiversity and better crop varieties. You must have seen, you must have studied crop selection. In crop selection, what we will do, we need to have resistant varieties, and we need to have better yielding varieties, and there will be hybrids created in between these two. So the parental stage we need to get from the nature itself, And industrial materials, the bulk materials, bulk is concerned, or whatever the things are there that we are getting from the nature biodiversity itself. And non-consumptive values, recreation and education, research, And traditional values, all these things are getting from the ecological services. Actually, in your exam, we have asked this question. And ecological services are nothing but these are all included in the ecological services. Balancing of nature. Let's see, there's a climatic seasons, rainy season, winter season, summer season, and there's a flora and fauna, balancing and flora and fauna. Everything is controlled by the ecological services. Biological productivity, regulation of climate, degradation of waste, cleaning of air and water, cycling of nutrients. This was asked. Control of potential pest and disease causing species. See, in the forest, there is no, we are not using any pest control like what we are doing in case of your fields. Still, the pest control are being managed over, diseases are being managed over there because nature is doing that one. Biodiversity is taking care of that one. Detoxification of soil and sediments, stabilization of land against erosion, then carbon sequestration and global climate change, maintenance of soil fertility. So these are the applications of biodiversity. These are the uses of biodiversity. This is one of the potential questions. So one could be different types of biodiversities in this one section. Okay, first could be from this section the different types, various elements of biodiversity, different forms of biodiversity that you should explain, and there's applications of biodiversity. And there is one more conservation method. Why the loss of biodiversity is there and how we can conserve. This is another question likely to come from this one. And measures of government of India to conserve the biodiversity. So biodiversity is very important, but now there is a huge threat to the biodiversity. Like for every hour, it is estimated to lose 3 species. So it is a high loss for the biodiversity. What are the reasons basically? Loss of threats to biodiversity. One is habitat destruction. Earlier we used to have forests, and even if you are having the paddy lands, paddy crops, and we used to have the banks, and there used to be a lot of trees, and birds used to form the nests over there. Now there are no trees, no. Even the lands are all made into small segments. They are not big lands. So the habitat is being destroyed like nothing. Even the forest also, a lot of the forest is being destroyed. So this is the one problem: habitat destruction. They don't have a space to construct their house. That's the one reason. Second, global climate change. Global warming is a big problem. It's not India or Hyderabad or Telangana problem. Globally wise, the change is coming. Not only the global, the temperature, but heavy metals, huge amount of heavy metals are being incorporated in the water and soil, and lot of pesticides. Earlier we used to have a deweeding process by laborers, but now what pesticide, the 4D sprays they are doing, that is creating a lot of nuisance. And habitat fragmentation. Earlier we used to have as a 10 acre for a farmer, or 15 acre or 100 acres also. Now they are small marginal farmers. It has been divided into sons and grandsons kind of thing. Now the fields are 1 acre, 2 acre, half acre kind of thing. So as the fragmentation has been taken place, then what happened? There is no scope of leaving the space for the biodiversity. Pollution is a one major threat because of the industrialization process, urbanization process. Huge pollution is leading because everybody are thinking of making life easy. Earlier we used to talk about only chlorofluorocarbons. Now radiation is a big issue, and radiation also comes under the this pollution. Overexploitation. Now we don't want to leave any space. Earlier, as I told you, they used to leave the space in between the land and in between the crops. But now there is no space. Everything we need to explore, even small gunta or small SFT space is there. And alien species. Now we are fancy about something new, something in US or something in Africa, something a new species if it is found. Like in your exam also, we were asked Prosopis juliflora, Sarcatuma. It has been got from African countries because it grows rapidly even in the dry condition. No water is required, no pesticides are required for this one. Diseases. Earlier we used to have very few diseases. As more mutations are taking place, potent pathogens are developing. So diseases are also coming. Plants are facing a lot of plant diseases. That's what it is creating a threat to the biodiversity. So these are habitat loss could be different reasons. One, human activities, cutting of the trees, and weather. There's another reason. Poaching. But not only the animals, plants are also being faced with that one. So this natural causes are narrow geographical areas, low population, and low breeding rate. Let's see, this elephants, they got very less breeding rate. Even your lions and the cheetahs, tigers, they got less breeding rate. So see, the production is less, but loss is very high. That's what is a threat to their biodiversity. Natural disaster also, natural as well as human disaster also, that is causing the. What we should do? So there is a threat. What we should do to overcome, to protect our biodiversity? What we should do for the protecting the intact? First of all, natural habitat, whatever the habitat is the place where the organism survives in ecosystem. If you take forest, in a forest, the area wherever the deer survives, that is a deer habitat. Try to maintain it as such. Try to maintain it as such. See, deers are very sensitive. You have to maintain its habitat as such. Otherwise, they will. In fact, if you catch hold them, even within an hour or so, they will die by themselves. They are very sensitive. Reduce human population. Say that is just yesterday, day before yesterday, it was a human population day, and there was a big debate how to control the population. That is, see, actually, population should be under control. Otherwise, if you are keeping on exploiting the nature, then what happened? There will be nothing on a day. And self-sustainable we should do. This thing, it should be available for us today, and it should be available for tomorrow also. Okay. And expansion of urbanization and industries. This is another problem. See, yesterday I have seen an article in Africa. The people are even from villages, they are going to the small towns. From small towns, the people are going to the big towns, capitals. So almost villages are being vacated. Here in in COVID situation, see, all sectors are affected. In fact, not much in case of agriculture. Agriculture is not much affected with COVID, post-COVID situation. So now, like we can say it's not reversed, but it is stalled. It is stalled for some time. So in African countries, even Malaysia or some countries, they are encouraging the people to stay in the villages. Some sort of remuneration or some sort of thing, and more industries should come in the villages, not in the. Generally, industries are located around the cities, but if you can have this industries in the villages, not the small rural industries, we should have big chemical industries and pharma industries also in the villages. Okay, and we should do the educating. This is the most important thing. Human population controlling. This, see, we'll say, but I don't know how much it is going to be worked out. And urbanization controlling, I don't know how much it is going to be worked out. But educating the people should be done. You should educate the people, and there should be there's a biodiversity village level units. There should be, and if somebody exploits the resources from the villages, and 1% of their benefit should go to the village. There is a village level biodiversity committee. I'll tell you one story in Guntur. Just 4 kilometers from Guntur, there is a small village. I forgot its name, and one company, I don't know, one company which produces this biopesticides. Biopesticides. These are the living organisms which kills the other bacteria. Basically, they have isolated one Pseudomonas species, one bacterial species from that area, from that village, and they are selling it in market. And they have earned crores of rupees from that bacteria, which is collected from Guntur. And these people have sued into court. They have the village people, and they have made a committee. There's a biodiversity village unit, it is called as biodiversity village unit, and they have sued into court. And as per the biodiversity law, Indian there's a biodiversity act, 1% of the profit should be shared with the area or the place from where you have collected the living organism, the biodiversity, or anything, a plant, animal, or pig, or anything that you have collected from that area, that should be given to that area. But the people who are surviving in that area, see, if you are collecting, there's some medicinal value from a forest. If you are collecting some medicinal value from a forest, if you are living in that area, you are free to use it. You are free to use it, and you are freely can give it to other people freely, I'm telling you, without cost. But whenever you are making a business, whenever you are making a big level business, then from the profit, 1% should be shared. Okay. So now 1.4 million named species are there which are known. From that one, 70% of them are invertebrates. And it is estimated that 3 to 50 million species are alive, but we have not yet identified. Earlier, if you see, we used to have this kind of habitats. Now still, some extent, this kind of platform is there in US. So they sprays the medicine, these pesticides and other things from the aircraft. But now it is similar to these small segments. Deforestation, huge deforestation is going on. See, earlier it used to appear like this, now this is. And causes because of commercial. In Andhra Pradesh, the situation is, don't say Andhra Pradesh, but here also. Some 75% of the genetic diversity of crab plants being lost in the past century. We used to, I don't know how many of you are aware about agriculture. We used to have Hamsa variety, Surekha variety of paddy. Hamsa, it used to come in 2 months, and short variety. And but we used to have the same pest problems and other things. But now we don't come across those varieties. Even we used to have HMT variety. Now it is there, but very rare HMT. But now this 1010, 1020, 1030 varieties have come. And this is the case of paddy, but this is same with all. The older varieties are being lost because we are depending on seed producers, which they are giving the genetically modified varieties. And this silencing technology. See, there's what they will do. They will genetically modified cotton, genetically modified brinjal. They all belongs to Malvaceae family. So what happens here? Pollen grain of one cross pollination is quite possible. So from brinjal, it will go to other brinjal or other tomato. It will do a cross pollination over there. See, it may not be fertile, but what happens? The gene, the gene responsible for silencing, that gets integrated over there. And what happens here? These whatever the seed that you are getting from the company, that gets germinates and it will give you a plant, and you will get a yield for that year. Subsequent year, when you are planting the seed which you got from this generation, what happened when you are germinating again second time whenever you are germinating this seed, what happened? The silencing gene gets activated. The silencing gene gets activated and it produces cyanide, and the plant gets killed. This is the reason many malvaceae plants of our area, wherever this BT cotton is being used, almost you must have seen small brinjal spiny brinjals used to be there in the villages. It's disappeared. Almost they are disappearing. This is the reason. This is the using of genetically modified organisms with silencing technology. Monsanto is responsible for all these things. And we are using it's the American company, and we are using Monsanto seeds, and even our Indian Mahiko is also doing the same thing. Okay. And the coral reefs also, let's say, a lot of biodiversity is under threat, and it is lost. So what we should do? Biodiversity inventories and conserving biodiversity in protected habitats. See how to in situ and ex situ methods are there. What is in situ and what is ex situ? We'll discuss those things. So in situ is getting all at one place and storing it. First, we'll see how to conserve the biodiversity. Restoration of biodiversity, and imparting environmental education and enacting, strengthening, and enforcing environmental legislations. We have population control, reviewing the agriculture practices, controlling urbanization, conservation of through biotechnology. So conservation, in situ and ex situ. So, what is in situ? Within that place, you are supposed to conserve. Wherever the rich area is there, wherever the rich biodiversity is there, in that area, you make some restrictions to entry of other organisms, like our human exploitations, so that the biodiversity of that area will be protected. Examples of sacred groves and lakes, and terrestrial area, marine areas, national parks. See, if something, if in one area more deers are there, or more lions are there, or more elephants are there, you construct a sanctuary of that elephants, or sanctuary of tigers, sanctuary of deers. You are constructing a national park of that area, or some park you are constructing, as such that the organisms of that area are conserved for a longer while. Here in the start of Medak Pocharam, we are having there's a sanctuary, deer sanctuary is there. It is a breeding center is also there. Even there is a small sanctuary for crocodiles also there. So, see, if you move the deers from that area to other areas, then there is a possibility of not matching of habitat, and it cannot be able to grow that easily. So, whatever the area they are living, make it suitable for them and let them survive in that area. That is the number one priority. Situation. Second, ex situ. What you need to do, you need to get the organism, plant, if it is in the position to lose its identity, get it at one place, keep it under our control or under our observation, so that it will be easily maintained. And seed banks, gene banks, cryopreservation techniques are also seed banks. we are having a rice research center in Malaysia is having a seed bank of paddy. The seeds of throughout the world, there's a rice seeds of throughout the world are collected in that area. And botanical gardens, zoological gardens, and aquaria. Botanical garden, different types of plants. It is mentioned that there's the plant of Himalayan, plant of Thar Desert, and plant of San Francisco. Like the plants from different areas, they are collected over there. And even zoo, you must have seen this tiger is collected from that area, and this lion is collected from that area. So you are bringing them at one place and giving a suitable environment so that they will see, even though they are getting over at one place, but they cannot breed as like sanctuaries it is possible. Number will increase more if it is in situ. But sometimes, see, botanical gardens and zoological gardens, not only the conservation, our aim is to recreation also, aesthetic look also, and to earn the money also out of it. Aquarium also. Aquarium is also a collection. If you go to the this Nehru Zoological Park, there is a big aquarium is also there. And this biodiversity is concerned, and distribution of biodiversity, the area where huge biodiversity is there, that areas are called as hotspots. Hotspots. And among the total world, like total world, there are two major hotspots which are located in the India. One is one hotspot is Eastern Himalayan region. Second is Western Ghats. There's a major. See, this is the one Western Himalayan region. This is Western Ghat. This is a Western Ghat. And individually, India-wise, if you take the biodiversity, we are having the 10 different zones, biodiversity zones. Let's not see detail into it. I think this is over with the first unit. Classification of living organisms, all these things you did in your first semester itself. And one yes, legal complications of biodiversity. Legal complications. See how to protect our biodiversity. India is having this question was also asked. We were having this the Biological Diversity Act, the 2002, which is dedicated for controlling or conserving the biodiversity. This is a very important thing for us there. It is there in the presentation. Where is culturable and unculturable? Is it there in the first unit or second unit? Yeah, it is there in the last unit. Okay. Legal aspect, the Act 2002 Act, I will explain later. Anyhow, it is like sections are there in that one. Section 1, Section 2, Section 1, like it is with prior permission. If somebody want to explore a plant or animal of area, Guntur, if you want to explore something, you should take prior permission from Biodiversity Board. The National Biodiversity Board should give you permission. Even this question was also there for you. You should take permission from there. And if you are applying a patent, see something new you got from your bacteria, you got a bacteria. You're as a microbiologist, you're growing. You got a bacteria from Nalamala forest, and that is giving you good antibiotics, new type of antibiotic, and it will kill even cancer, neoplasia cells, cancer cells also. So for that one, you want to get a patent. So what you need to do for research and for self usage, you can get unlimited material from that area. You are doing academic research, you are a student, I am also a teacher, so I can get unlimited material from that area and I can work. When I want to explore it for commercial purpose, even when the farmer wants to use it, even it must be grown in his field itself. When he wants to use it for a large scale commercialization, then he should take, when he want to take a patent, he should take a permission from the National Biodiversity Board, and he should pay 1% of his benefit. It is called as royalty. 1% of his benefit to that development of that biodiversity. Not the people of that area. It is generally mistaken. Simultaneously can be given for the people also. When it is coming for when you are collecting a tree or suppose bacteria or a gram soil from Nalamala forest, so the farmers, if you can give the money to the farmers, they will prevent, they won't cut the trees of the Nalamala forest. Indirectly, you are benefiting to the Nalamala forest itself. Okay. So this is about Biodiversity Act. Penalties are given in that one. If somebody breaches this act, what should be there? This is also like important for short answer question. Biodiversity Act also. Next, unculturable bacteria. That's microbial interactions. We'll see later. Microbial interactions. What is how we should understand the biodiversity. One method I told you, comparison of sequences. Different methods are out there. How to compare the microorganisms, how the biodiversity should be compared. One is based on their morphological characteristics, like how the cell is there, how the if species genetical biodiversity is taking dogs. How do you compare? How their ears are there, how the feathers, hairs are there, how the body buildup is made. Isn't it? That is external morphology. And for this one, you need to have a suppose because we are working with microbes, you should have a pure culture for it. When you want to compare the morphology, you should have the pure culture in your hand. Okay, and microscopic characteristics also should be compared. Chemical composition, like what the cell wall is made. What the cell wall? Now it is the fatty acid, methyl. There's a fatty acids of cell constituent is considered to be the highest level of characterization. So this is external morphology. This is a colony morphology. It could be circle or irregular or filamentous, rhizoid, and from the top it could be entire or undulate or filiform. Different types of colonies you'll be getting. And microscopy, it could be gram positive or gram negative, and it could be bunches or diplococci, and here it is a capsule. This is a capsule here, and like how it is showing in the fermentation pattern. Yesterday, your senior subject fermentation, this biochemical test results, indole, methyl red, August piscus, Siemens citrate results they have kept. So how the chemical reactions it is showing, that's important. See, the first you can see this is an EMB agar. EMB agar. Here the crimson green, shiny green colonies you will be able to see. If there is a shiny green colonies, that is the E. coli, typical E. coli, and this is a gram negative small rods. It is a E. coli again, and this is a Staphylococcus aureus, and this is growth on the mannitol salt agar. And the cultural characteristics, how the bacteria are growing, what nutrients they are taking, and what temperature they are growing, and whether the oxygen is required or not, that is also considered for the classification. And Antigenic characteristics also we will consider. See, if the more the closely related, they will be sharing the antigen antibodies. See, woman shares the antigen, the blood grouping antigens with the monkeys. Rhesus, there's a RH factor is there in man as well as rhesus factors. So they will be antigenically similar, means they are closely related. Genetic characteristics, DNA RNA comparison. See, DNA comparison we discussed. See, different methods are there. Pulsed field gel electrophoresis. Gel electrophoresis you must have seen. See, a gel is prepared. In that one, wells are there. In the wells, the DNA is loaded and it is separated. Whereas pulse field gel electrophoresis, what we'll do, we'll be providing the electric shocks with a 40 degrees. See, here, once you will be giving shock, the current from this direction, then it will be from this direction, then is this direction, then this direction. So every time 40 degrees change will be doing. So here the advantage is here not only the molecular weight, based on their conformation also we can separate. Based on the GC content also we can separate what you call your DNAs. And then is the different AFLP, amplified fragment length polymorphism, or RFLP, rapid fragment random fragment length polymorphism. I'll show you here. It's a restriction fragment length polymorphism. See, this is the wild variety. This is the wild variety, and here restriction one X bomb H1 cuts here, here, and here. So if it is cutting here, here, and here, you are getting two fragments. And here I am using the probe. That's what the radio labeled fragment is going to be larger fragment. Here, this is going to be this much fragment. Okay. In a variant, genetical biodiversity containing other variant, here what happened? There is a mutation at site 2. This bomb H1 is no more cutting here. Is no more cutting here. So what happened? The radio labeled probe is now much bigger than that earlier. Now it used to be only this much fragment, but now it is a very big fragment. This is called as restriction fragment length polymorphism. Restriction fragment size is getting changed here. Simple. This is very much useful to diagnose your criminals, maternity, paternity detections, and such kind of detections. It is very much useful. And here the problem is, see, suppose you are trying to identify a criminal. Restriction fragment length polymorphism is simple. You will be taking the DNA from the cell and you will be cutting with the restriction enzymes, and you will be separating on the electrophoresis electrophoresis. But when you are collecting blood sample from a crime scene, the DNA is going to be very less, isn't it? And the DNA is going to be very old DNA, and you may be getting very little genomic DNA from it. Isn't it? You may be getting very small DNA fragment from it. So what should do? You should do another technique called as a amplified restriction fragment length polymorphism. AFLP it is called as. What you need to do, first we will be cutting the small fragments. See, it is cut into small fragments, and after cutting, you will be keeping the adapters. See, small DNA is added to it. This is called as adapters. Now you design the primers to these adapters. Now you amplify your amplified fragments. What about the cutted digested fragments? So even though small DNA, 1 or 2 DNA cells are obtained, that is bound with the adapters and amplified into large fragments, huge size, and they can be easily separated on the gels. The radio labeling techniques, see, this AFLP is a technique to increase the sensitivity. See, if you are separating in electrophoresis, if you are separating 1 DNA or 2 DNA, you cannot see. If you are having 1000 DNAs, then only you will be able to see. So, similarly, this radio labeling technique is one of the advanced technique to improve the sensitivity. See, radio labeling, radioisotopes are the elements which emits the radio waves, alpha, beta, gamma. They will emit. So this can be, when they emit the radio waves, they can be easily detected. Huh. This is okay. Anyhow, radio labeling, you must be aware about carbon dating. Carbon dating. There's instead of C12, we'll be using C13. C13. So we know that there is a half-life period. 60,000 years is the half-life of carbon. So we'll compare how much C13 is remaining in that one. Based on that, we'll be able to find out the age of that tree or the fossil, whatever that is available for us. And this is sequencing. For sequencing, what we'll do, this is an end termination or D-dimer method or Sanger's method sequencing. What we'll do here, see, we'll take 4 tubes. In 4 tubes, 3 the nucleotides are common, are normal one, normal deoxynucleotides, whereas one, see here, ATGC are there. In this case, what they are doing, adenine is going to be 75% adenine is going to normal, that is the deoxyadenine. 25% adenine is D-deoxyadenine. It is going to be D-deoxyadenine. So what happens here? Whenever the D-deoxyadenine comes, there the reaction stops. It will not, because there is both 3' as well as 5', both of them are having only H group over there, not hydroxyl group. So if you want that PCR to continue, you should have OH group. Okay, this is the method of sequencing. So sequencing, you are supposed to compare which sequences, as I told you, 16 sRNA for bacteria, and in case of lower eukaryotic microorganisms, it is PTS RNA or IS elements. Whereas in higher organisms, what we should compare? It is cytochrome C. You are supposed to compare cytochrome C. And see the protein, by comparing the proteins also, there's one X protein, cytochrome C. You take cytochrome C and you do a sequence analysis, and you can find out the relatedness among the 2 organisms. And fail, as I was mentioning that membrane fatty acids, methyl esters. What they will do? These are the fatty acids, and it is reacted with ethanol in the presence of catalyst. So what happens here? Glycerol is formed and fatty methyl ester is formed. This is methyl ester is formed, and this is separated by the gas chromatography. And these fatty methyl esters are unique to the bacterial type. Even strain level differences we can identify by doing the frame analysis. This is the most advanced technique in case of bacterial identification. So that is identification of bacteria, culturable and unculturable bacteria. Unculturable bacteria, how we should identify? We don't get pure culture there. There is no chance of getting a pure culture. So genetical level DNA sequencing we can do, and we will try to identify. Membrane fatty acid analysis we need to do, but there is a possibility, if you are getting a soil sample from a field, then there is a possibility of different bacterial cell wall in that one. So there is a every possibility of contamination. So how to eliminate it? You need to do a dilution. You need to keep on going a dilution. So till end point, it is called as end point dilution. You need to do the dilution, and once you are confirmed that only one type of bacteria are there, then you are supposed to go for a fatty membrane analysis or sequencing analysis or antigenic analysis. That is for uncultured bacteria. There is a one small section in your second last unit. That is a great plate count anomaly. Actually, there are more number of bacteria, almost 99% you can say, they are unculturable. Only 1% of the bacteria are culturable. What is unculturable? We cannot grow this bacteria in the laboratory conditions. We cannot grow this bacteria in the laboratory conditions. As per the Robert Koch, he said that every bacteria, every pathogen can grow. Even all the pathogens also cannot be grown in the laboratory conditions. We are trying our best to grow them, but we cannot grow. So you get a soil sample from a fertile soil, a garden soil, and you do a dilution and put a drop of this diluted water in a newborn chamber. This, and observe under the microscope and count the number of bacteria in that one, or hyphae or whatever the living forms, microorganisms you count. And a similar sample you plate it on the nutrient agar plate. Nutrient agar plate. Here, in case of nutrient agar plate, hardly you will get 1 or 2 colonies, whereas the count is around 100. So, by this experiment, we confirms that this actually we are supposed to do the experiment also for you second semester. So it confirms that majority of the bacteria which is there in your fertile soil sample that is not growing in the laboratory condition. This is called as great plate count anomaly. This is called as great plate count anomaly. What are the reasons? It could be, see, there should be a signal which is there in the which is contributed by the soil there, or could be the signal by the environment, or could be a signal by the living organism itself, that is called as quorum sensing. Quorum sensing. So that is missing in the laboratory conditions. That's what these organisms are not doing. So 99% of the bacteria which are there in the soil that cannot be cultured. 1% of the bacteria which are being cultured, and among this 1%, again we have exploited only 0.1% only. Is a 1%. Among 1% and 1%. That means total from the soil, 0.1% of the microorganisms are only exploited. And there are two short sections, preserved ecosystems and perturbed ecosystems. Preserved ecosystem is the ecosystem which is maintained constant its features for a very long period. Pond ecosystem, whatever you study, about 50 years back, even now, it is almost same. Whatever the zones are there, littoral zone, limnatic zone, and the benthic zone, it is going to be common. Always, it is to be the common. So, see, maybe this littoral zone may be converted into a terrestrial zone, and the limnotic zone may be converted into littoral zone, but it is called a succession. Succession may be happening, but overall, see, benthic zone may be decreasing, but overall, see, even though it is decreased, but if you take a pond ecosystem, these zones are going to be there always. The floating trees are there in the pond ecosystem, submerged trees are there, and there's some fishes or some planktons are also there in that one. This is going to be constant for a very long period. Isn't it? The zones are very much, and organisms are also almost I'm using almost they are same. Okay, this is a conserved ecosystem is concerned. Whereas perturbed microbial ecosystem, the best example is Antarctica. Antarctica is a glacious area actually, where the completely snow is there, where actually more of lower organisms are there, more of microbes are there, more of small invertebrates are there. Like you cannot expect much trees and much larger, maybe some there's a wolf, there's a polar bears, such kind of may be there, but they are less when you compare with other organisms. So when you compare few decades to study, many organisms are getting disappeared because of human exploitation. One is human exploitation, second is global population, global warming, and the ice is being melted over there, and due to ice melting, the area of the terrestrial area or the snow area is getting reduced, and it is human act, because of human are going there and doing research, lot of research for exploiting commercially important microorganisms. See, like in Western countries, the temperature is low. So we need a bacteria which produces amylase, protease, lipase, which can work at low temperatures. Isn't it? Then only you can work at, your detergent will work at low temperature. Because of these exploitations, see, these antibiotic resistant organisms are developing over there, which is huge. Antibiotic resistance, if you are getting, then a lot of normal flora will get killed. And there is one more topic for you: the microbiome, plant microbiome and human microbiome. What is microbiome stands for? Microbiome is some total of microorganisms associated with an organism. Here, some total of microorganisms associated with man is called as human microbiome. Or second, see, so because of molecular biology, the people also have defined the sum total number of genes of microbes associated with man is also called as human microbiome. Whereas plant microbiome, some total number of microbes or microbial genes, genomes associated with the plant is called as plant microbiome. See, this is a philosphere. In fact, we will be doing this experiment. If you take a leaf and put it in nutrient agar, you will get this kind of culture. A lot of microbes are growing there. So, how this microbes are associated with plant and man? In case of plant, they promote the growth. You know, you must be aware of Buckney disease, Gibberella fujicora, foolish seedling disease. It is called as. It will grow. Cytokinins, auxins, gibberellins, auxins. Your E. coli produce auxins. You know, it is the indole test. Whatever the biochemical test we do, it is the indole. Indolistic acid is auxin. So E. coli produces auxin. So these plants do promote the bacteria, this plant growth and mitigate abiotic stress. So the stress will be relieved from the plants, and they work as a defense. See, when they are covered on the leaf and the acid rain is happening, then it is the microorganisms which will prevent the leaf from the loss. Okay, and these are the things will, microbes will create a systemic resistance, and they will control a lot of diseases. Microbes will control a lot of, all the microbes are not disease causing. Majority of them are helping. So what happened when they are making a layer on the leaf? When the pathogen enters over there, comes over there, they have to face first to this good bacteria. So the prevention of disease is possible there. Okay. So sometimes directly or indirectly they will help in the controlling the diseases. That is on the top. At the bottom in the soil, see, they will mineralize a lot of minerals. See, solubilization is required. Iron solubilization, phosphorus iron. For iron uptake, citrospores are required. Citrospores, see, plant directly cannot take iron. So citrospores, for that one, microorganisms are must. And nitrogen fixation, you know how much important is there. And rhizobium bacterium. And a lot of mineralization process also there. And nutrient uptake, nitrogen fixation, all these are the activities which are helpful for the plant microbiome to the plant. And this is inoculating of microorganisms we will do for improving. You must have heard about biopesticides and bio inoculants. For rhizobium, generally what we'll do, just along with the seeds, rhizobium is mixed so that when the plant is as soon as the groundnut germinates, This microbiome comes in contact with the roots and makes the root nodules, and there it does the nitrogen fixation. That is application of individual organism. Whereas sometimes you need to apply consortium, not a single organism. You need to apply a group of organisms, not single organism will do this activity. So this is called as microbial consortium. See, when you are looking for biopesticide activity, it is not single organism we use. Generally, Bacillus subtilis is used, Pseudomonas is used, Trichoderma ricci is also used. So, whenever you are applying the organisms, see, it will be establishing, it will be colonizing on the plant. So some poor organisms will be there, and around the poor organisms, other organisms. See, the poor organisms are not beneficial to the plant, but they are required for the pollination of the bacteria, healthy bacteria or good bacteria over the plants. Okay, these are likely to come in the short answers. The human microbiome, lot of microorganisms are there on and inside the human body. On and inside the body, there are huge number of microorganisms. In fact, we cannot survive even for a minute without microorganisms. We cannot see. We first feed is the milk, and in the milk we got lactose, and there is no mechanism in our body to consume to digest this lactose. So what happens if you are not able to digest the lactose? Then what happens? It will be coming in the form of vomits. So there should be an E. coli inside the intestine to digest this lactose. So you take different levels of body. We are having different types of microorganisms, and which will be giving the protection to our body. It's not going to detail. It's the interaction. Where is the microbial interactions presentation? Okay, I will do from the textbook. And one more important long answers that is likely to come is microbial interactions. There is only one thing remaining. Yeah. It will take 5-10 minutes. Just brief I will do, because everybody, you must be aware about it for a very long period. So microbial relations, see, we can study the interaction within the single population as well as in ecosystem. In a single population, we can take the example of the culture which is being grown in a flask. It is called as batch fermentation. It is called as, it is a batch fermentation or closed fermentation, where the pure culture is being grown. So what happens here? There is a positive interaction. There is a positive interaction and negative interaction. Positive interaction is possible in the early stage of growth, and negative interaction is possible in the late stage of growth. In the beginning stage of batch cultivation, what happened? The enzymes, hydrolytic enzymes are produced, amylase, protease enzymes are produced by the microorganisms, and they will release it to the medium. So they will digest the media. So this digested food will be consumed by the other bacteria also. So this is the positive correlation. In negative correlation, once the bacteria goes to the stationary phase, the nutrients are about to exhaust. Isn't it? Nutrients are about to exhaust, then they will compete, they will fight among themselves for getting a nutrient. Second, there will be a waste accumulation. there will be waste accumulation over there. So this waste created by one bacteria may be harmful for other bacteria. So like this, there will be within a batch fermentation, there will be a positive and as well as negative relation in the same population of microorganisms. Okay. Now interaction between the different microbial populations. Microbial populations you take, we have different types of associations. It could be neutralism. Neutralism, both the populations are neither benefited nor lost. Commensalism, here one population is benefited whereas other population is neither benefited nor lost. Can you give an example of commensalism? Commensalism. Are you able to hear? Arvind? Gautam? Hello? Gautam? Hey, Gautam? Nikhil?
Yes, sir.
వినపడుతుందా?
Yes, sir.
Okay. Sorry. So neutralism and commensalism is one benefited, whereas the remaining is okay. 5 minute class हो गए. And commensalism and synergism is both are benefited. Like most of the microbes in the soil, they are synergistic. They will perform the life in the synergetic way. And mutualism, there is one more here. It is symbiosis. You can take example of rhizobium with leguminaceae plant. Leguminaceae plant will give a shelter and as well as metabolites to it, whereas this rhizobium will do the nitrogen fixation and it will provide to it. Competition, there will be a loss for both, and amensalism, one will be loss, other could be benefit or no. And in case of predation or parasitism, the predation is taking away the thing and going in some other place and eating over there. Parasitism is sitting on the host or going inside the host and eating it. So it is parasitism. Commensalism examples are chlorella and which increases the growth of Pseudomonas and Xanthomonas. The substances which are produced by chlorella will be increasing the growth of Pseudomonas and Xanthomonas. This is one example. This is for and synergism. Both are benefited here. Here Pseudomonas turgii, here it can cleave there's a paranitrophenol, but cannot use it. Here, whatever the substrate, whatever it is being produced, that will be utilized by Pseudomonas aeruginosa. Okay. This is synergetic relations. This is mutualism. This is also beneficial. Both are getting benefited. Rhizobium is an example, and paramecium, in paramecium, chlorella also stays over. This is the question which was given to you in exam. In paramecium, endosymbiont, we asked what is the endosymbiont. Chlorella is an endosymbiont with the paramecium. These are the examples. And legions are one best example for symbiotic relations. They cannot survive individually, separate. Okay. Here the mycosymbiont as well as cynosymbionts are there. Cynosymbionts are the algal partner. Mycosymbiont is the this fungal partner. And this if you separate these two things, they cannot survive. This if you try to grow this with the fungal partner or algal partner, you cannot grow them in the laboratory, whereas they can grow easily. And even was a question from this section also. These are the indicators of pollution. If there is a loss of, if there is a huge pollution, you will be getting a there is a possibility of disappearance of lichens of that area. And lichens are responsible for rock weathering. And so they're the beginning step of succession of rock ecosystem. So if you want to convert rock to desert to soil, so this lichens works very important role, and a lot of compounds they will produce. Because medicinal value containing compounds are also produced by the lichens. This fluorescence also, one question has come. Luciferase is an enzyme which is responsible for the fluorescence. And this ruminants is a best example for symbiotic relations. This is actually, as I told you, not only the human being, even cattle also cannot be able to digest this cellulose. So there's a cellulose digestion, fatty acid creation, and these activities are done by the microorganisms. Methane generation, fatty acid production, even digestion of cellulose. This is done by the microorganisms which are there in the rumen. Negative relations are concerned, amensalism. So here what happens here? Negative microbe and microbe interaction here in this case. Here, siderospores, it is sometimes useful, but if some bacteria produces more siderospores and it acquires the entire iron. So the iron is not available for other organisms. Sometimes antibiotics are produced and these antibiotics kills the other bacteria. Parasitism, they goes and infects other bacteria. They could be ectoparasite or endoparasites. This is there in the book. Facultative and obligate. Facultative, if a pathogen will survive as a parasitic as well as non-parasitic life, it is called as facultative. If a parasite survives only in the parasite stage, it is called as obligate parasite. Mycoparasitism, if a fungi is infecting other fungi, it is called as mycoparasitism or super parasitism. Predation, there is something taking it away and eating. It's already I discussed. Understanding microbial diversity with culturable bacteria. Okay, this is how you end. Just go through the entire thing, and if you have any doubts, not only this, I will explain you if something is required. Okay, with this, because principal says visit is there to the department, we'll have some work to do. So I will end the session now, and we'll see. Like if the time permits, tomorrow I will do the first semester revision of first semester. Also, I will try to do tomorrow. Some other time at least.